We saw, this randomized control trial is not an easy thing to do, and there are lots of procedure, systematic procedure to be followed. And depending on the nature of the study, whether it is therapeutic or preventive, whether it is double-blind or a single-blind or open trial, say the name of the trial is indicated with all those characteristics. We saw that yesterday. Say now our study outcome could be a quantitative outcome, like say reduction in the blood glucose level, or a qualitative outcome, say feeling better, quality of life measurements. And we can may have a primary objective, primary outcome. We may be interested in looking at the efficacy of a drug, which would be our primary interest. We may also have some secondary outcomes to see, the level of toxicity, and there can also be multiple outcomes in a trial. So you may be interested in seeing with the efficacy level and also at the feasibility or acceptance level. See, when you are dealing with multiple outcomes, we need to be careful. See, we should rather have too many outcomes. Sometimes what happens, one outcome, if you are interested in it, it may have an impact on the other outcome. To give you an example in tuberculosis, see, suppose if you are interested in the efficacy of a particular drug, that's your primary one of the outcomes. And also you are interested to know the acceptability of that particular drugs to the patient. So you have two primary outcomes you have. What we will do, suppose our outcome is to see the efficacy of the drug. So we will try our best that the people, they take the drug. If the people are defaulting, we will send a health visitor to their house, motivate them, give them. So our aim is that as many patients as possible should take the drug. Then only we can give a good estimate of the efficacy of the drug. Okay. In that process, what we are doing, we are rather foreseeing, say, the feasibility aspect of it is hampered on that. Suppose, we are interested on the feasibility, we say, okay, I want to rather look at the feasibility. If the guy comes and takes, then only, the medicine is taken. I am not going to take any special efforts. What happens? Only about 60-70% of the people, they take the drug. You can't rather give a good estimate of the efficacy of the drug. So, when you are having two outcomes, one is, if you give emphasis to the one, you can't measure the second. So when you're choosing your multiple outcomes, you'll have to rather see in such a manner that they don't affect each other. Okay? There could be some intermediate endpoints. So you may look at, say, the change of, say, for CD4 counts, and then you may still proceed on the trial to rather look at the other outcomes. So in a study outcome, there could be different points which, of course, have to be stated well. We will be seeing, say, I will be seeing something on the protocol also for RCTs. So mostly we deal with phase 3 trials. So any examples or any sort of a thing that we give now, are essentially for the phase 3 trials. Phase 1 and phase 2 are essentially to see the safety, and it's done on a very small number. Phase 3 trial is the real trial to rather look at the efficacy of a particular drug. A trial could be a comparative or control trial. What do you mean by that? Suppose there is already a potent drug available and you are going to test another drug which is more potent than that. It is a comparative trial like. If you don't have anything, then you go for a placebo trial. Okay? So variations are kept under control. See, this is one of the important aspects of RCT. We try our best. That's why it's an experiment, intervention, which is contrary to the observational studies where you allow things to vary in the nature. Here you are controlling them. Okay? And you see to it that the group, everything is similar except the intervention. In fact, the number of times of your assessment, number of times they come to you, number of times you go to them, everything is standardized. You do it on an equal basis, similar basis to both the group. The only difference is some have the drug, some have a placebo or a control. And lots of statistical ideas and methodology are integrated into the RCT analysis. You have huge books on longitudinal data analysis, because what happens in an RCT is there could be some dropouts, withdrawals, all those things have to be adjusted in your analysis. And so there are a number of excellent statistical developments are there in this field on longitudinal data analysis. And elimination of bias is done at the design stage. Bias have to be tackled at the design stage. You can't rather eliminate a bias at the analysis stage, though some measures can be rather used to do that. So what are the interventions in an RCT? Lifestyle, change in lifestyle, in CVD risk factors. RCTs, you can rather give health education talk, and that could be an intervention, change in the lifestyle. And then you can rather observe them, people with intervention and without intervention. Diet, drugs, some operational factors, surgical procedures, rehabilitations, any of these things can be intervention factors, but in an RCT there should be some intervention. There should be some input. Then only it's an RCT. You are not observing without any intervention. Specific issues of, some of these things may be repeating. I might have rather said the same thing earlier, but it's all a question of reinforcing the issues. The issue is so important, it's worth repeating them. Okay? So the specific issues in RCTs are sample selections. How are you going to do a sample? Most of the time what happens, RCTs are not done on a representative samples. It is done on, say for example, TRC studies and all, were done on patients who are attending the government chest clinic, which is situated in the Chetpet. Okay. People who come there for treatment, if they qualify or if they are eligible for certain conditions, they are transferred to TRC for studies. So what happens, it is not a representative sample of all the cases like. It is a specific case who come to that particular hospital. Okay? So RCTs in that way may not, let's say, you have to rather take care of that when you are generalizing your result later on. Okay? Sample size. Adequacy of sample size, I think most of you, senior enough, I think you might have rather come across in your life when you are doing some study. People say, you must have minimum. So I can't just study 2 patients and then generalize that in my results. Okay? I need to have some sufficient number of cases to work. So there are methods of finding out what should be the minimum number of people you should have in both the groups, your intervention group, in your non-intervention group, so that you can get some valid conclusions out of them. We get a lot of dentists coming to us for the consultant, and they say, most of the dental interventions, are very expensive and they can't do on many people. And it's usually the people who come to us are MD students, and they have their own limitation and they have to complete it within 1 year, 1.5 years, and then say, my resources are limited. So we clearly tell them, yes, we understand your position, but this is a problem. If you do it only with 10 patients, then you may miss some important conclusions on it. Okay, that is, so sample size, we can have a separate session on sample size, how to rather go about it. Okay, choice of control groups, how we are going to rather do this control, and what sorts of randomization procedures, how are you going to go about with your blind assessments, are you going to rather make it rather single blind, double blind, and what is your length of follow-up. These are all very, very specific issues which you have got to be terribly clear before you start doing your RCT. See, sometimes the true effect may not be ascertained for many years. You may need a long, say for example, exposure to certain carcinogenic agents. See, you may rather find the nuclear, this thing, you may get an effect after 15 years or 20 years. Okay? And study power is dependent on the number of events observed during the study. And random allocation, say most of the case, the random allocation is easily said than done. When you take a journal and read, many studies, they will just say we randomly selected 150 people and we studied this. Okay, they will not give any other detail about that. You ask them how you select this random, and ultimately you will find that it is not random. The random is very loosely used. So the random sample has got a meaning as such. They may probably go and then they say everybody who comes between 10 and 12 in the marketplace, we picked every third guy. So it is a random sample. It may be say they all late sleepers and the people who get up late are missed in this whole sample. Because you are going at 10 at the marketplace and people who still are at the bed, you don't catch them. Okay? So random sample has got a real, see, people, random unfortunately have got two meanings. In the general saying random and then the statistical sense random. What we mean here is it's in the statistical sense, it should be a random allocation. So now let us rather say, what are the essential steps? Suppose if you are going to do an RCT, okay, just try to rather put in a hierarchical form, what are all the things that you need. First of all, you should define the purpose of the trial. What's your research question? What do you want to rather study? Say, suppose you have a new drug for a particular disease. I want, and then it has gone through phase 1, phase 2 trials. It has been seen safe, and there is a claim from a pharmaceutical institute, a company, that it is a very potent drug. Okay, so that is your research question here is, I want to see whether this particular drug is superior to the standard treatment that's available right now. State your purpose. Then design the trial. Design the trial, as I was rather telling you, different steps that are required. You have to write a protocol, a detailed protocol, work instructions, standardizations, what sort of a test you are going, how are you going to do the screening, what is your eligibility criteria, what is your inclusion criteria, all those things you have to rather write. And so protocol in RCTs are considered as a very sacred document. No one has got right to meddle with it. It's like a constitution. Okay, you can't rather do an amendment of constitution unilaterally just like that. It has to go through a procedure. Okay, so protocol, once it is finalized, it is a sacred document. Put all the details, minutest details, into the protocol. Then you conduct the trial. In conducting the trial, you've got to be extremely organized. Conducting a clinical trial, because every stage you have to document, especially nowadays, your trial is subject to clinical trial audit. Okay, all your procedures, any minute change that you make has to be documented, and it is subject to verification by a general body. And stopping rules. When are you going to do interim analysis? State them in your protocol. Analyze the data. Here also, you do a descriptive and what are the tests of hypothesis that you are going to either do, draw conclusions and publish results. See, this is the broad, just rather put in one slide, the whole steps of doing an RCT. It is not that simple. Okay? I will just try to either tell very, very briefly about this GCP. GCP are the International Ethical and Scientific Standard, setting the minimum requirement for the development, conduct, performance, monitoring, auditing, recording, analysis, and reporting of clinical trials that involve the participation of human subjects. To put the whole GCP in a nutshell, this is what GCP is. So the International Conference on Harmonization, in fact, they are the one who had come out with this GCP guidelines, okay, which are, say, the US Food and Drug Administration, that follows that, the guidelines for clinical trials on pharmaceutical products in India, Central Drug Standards Control Organization, DGHS, Ministry of Health and Family Welfare, Government of India. They all rather have subscribed to these GCP norms. And this GCP synopsis as such is the regulations for informed consent. There are regulations for institutional ethics committee, defining the responsibilities of the sponsor and the investigator, control of investigational products, required elements of the investigator's brochure, and they have given the guidelines for all the essential documents which has to be meticulously done. So now, I have about 4 or 5 points on designing a controlled clinical trial. So the starting point for a controlled clinical trial is a clear statement of research question. Okay. It's always advisable to have a single primary research question. I was mentioning about this. When you have multiple questions, be careful. But there can be secondary questions also. And the trial will be planned to answer the primary question with adequate power. How many of you know the value? Say the power doesn't mean your muscle power. Okay. The power is a statistical power which says, if there is a significant difference, if you want to understand it in one single way, sentence, if there is a difference, you should be able to pick the difference. That's power. If the power is 90%, then I am 90% sure of picking up the difference if there is a difference really. Okay, if the power goes down, what happens? I may miss to pick up a difference. Okay, so adequate power, you should be able to pick up the difference. The second is the primary research question will determine the study objectives. And that will help us to set up appropriate hypothesis for evaluation. Your research question will be a general question. Okay, is this statement better than that? Okay. Based on that, you set your objectives, and that objective will lead us to setting up a statistical hypothesis. So the next step would be to prepare a well-organized written protocol for the clinical trial. And the protocol contents are study background. You would rather see the selection bias. We all rather understand, the selection bias as selecting, in your sample if there is a bias. Inadequate literature search is also called a selection bias in RCT. Okay. Inadequate literature search is also considered a selection bias. You have not done your literature work well. So you have got to do a good literature work, and that should be reflected on your study background. And then statement of objectives, your primary objectives, your secondary objectives. Then the third stage in the protocol will be your study plan. And study plan is the study design, and that should permit valid statistical inference. Describe patient and control groups with rationale for choice. Is it a single center or multi-centric study? Sometimes what happens, you can't get from one center the adequate number of cases. So you plan multi-center trial so that it can be done in different points of the parts of the place and at the same time, and so you have a pool of cases from various centers. But what are the potential difficulties? There is a center in Chandigarh, there is a center in Chennai, there is a center in Thiruvananthapuram. What are the potential difficulties like? In a multicentric trial, extra care need to be applied on standardizing. People there should use the same reagents, same sort of a clinical procedure they should adopt. Everything has to be standardized to such an extent that otherwise, you may get totally distorted pictures. If people in Thiruvananthapuram does on their own way and people in Chandigarh does on their own way, you can't pool them together. See, you are pooling them together because they are all completely standardized. Then only you can pool them together. So, yes. So, to do a multicentric study is very difficult. There should be a monitoring unit, a central monitoring unit. In traditional medicine research, there is a central biostatistical monitoring unit situated in Chennai. And they do site visits to see whether the randomization procedure is followed. Adherence of protocol is one of the very, very important aspects. The monitoring committee periodically sees, and patient inclusion and exclusion criteria, method of randomization and blinding, and study subject withdrawal. These all comes under the study plan in the protocol. Okay. Then the study drugs. You have to either give a fairly good description about the study drugs. What such of a drug you are going to use, what sort of a method of dispensing, method of administration, how are you going to rather make them look similar. Okay, Central Drug Research Institute, Lucknow is the one which has been rather doing a lot of standardization thing for us. And at the end of it, they show a capsule which looks exactly similar, so you should not be able to rather differentiate it. All those things, and suppose certain vaccine vials, you'll have to rather dispense it for 3 people together or something like that. You can't, once you dispense to one person, you can't throw it out. See, there are certain things which are which have to be given for multiple people. Okay, once you take it in a vial, all those things. What's its method of administration? How you are going to do about it? Any concomitant medicines or procedures. Say there may be a main drug, but you may also want to give a vitamin tablet or something like that as a concomitant medicines. That should come in the protocol. The 5th point is the measurements and observations. Okay. What is the response variable? Is your measurements valid and reliable? Say, suppose you are doing an intervention on a nutritional intervention, and you want going to rather measure the change in weight or height, and okay, and you have standardized anthropometric measurements and things, and all the equipments you have standardized. And your measurement, you have to rather see how valid and reliable your equipments are, and then the measurement technique. Height, if you put the rod like this, you can have one. If you put the rod like this, you have another. See, there can be a lot of variation in measuring heights. That variation may be much larger than the variation you expect because of your intervention. Okay. So you have to be very careful whether you have to really standardize the way it is being done, and you ensure that your measurements are reliable and valid. Measurement schedule. How I am going to rather do the measurement? What is the frequency of my measurements? Okay. And is there any surrogate response variable? Sometimes it is very difficult to measure the actual response variable, but you have a surrogate response variable to rather see. CD4 counts is one of the examples you can say. Intermediate endpoint like CD4 in AIDS or mortality in HIV. See, what happens, the mortality in HIV positive reduces the period of follow-up. Somebody dies, you want to rather follow up for 5 years, they may die, and say this sort of an attrition, you have to account for them in your analysis. Okay. So the impact on sample size, that's what, what is the impact of this sort of an attrition on your sample size? Should truly reflect the clinical outcome and be acceptable to the study patient. You have got to be very cautious about that. And in fact, this evening I am going to talk on the evidence-based medicine, and we'll be seeing some aspect of this. See, so adverse effects of intervention may be incompletely evaluated. Say this is again, people may, you have to document any adverse effect very carefully. It may be a mild, some people may say headache, but that headache also has to be documented. Sixth point in it is the statistical methods. How are you going to rather do? There should be a detailed section on sample size, how you have computed your sample size, and how are you going to do the dropouts, missing data, what are the covariates that you are planning to measure, are you going to do any subgroup analysis? This has to be planned earlier. After the study, you can't rather say, I am going to do this sort of subgroup analysis, because your trial sample size may not be adequate to do a subgroup analysis. Think of it. That's why, in RCTs, you do a lot of thinking before you start your work. You don't rather work and think. You think and work. And when are you going to do interim analysis? And there should be a very clear rule on terminations. In many of the, say, if my results are like this, I am terminating this project at this stage. The trial, okay, either adverse effects or you may have an overwhelming response of the new drug, you may stop, okay. And what sorts of analytical tools are to be used? You can rather mention that. 7, adverse effects reporting. Adverse effects could be a clinical or laboratory supported stuff, okay. And the institutional review and ethics committee approval. And in this type of RCTs, it is not just one time they sit and they clear. Usually the ethics, they have a review meet, and they have a periodic check of all your procedures. And I will just rather put few ethical points, issues in the clinical trials. The ethics of doing and not doing a clinical trial, the first and foremost thing that they look at it is no harm to patient and no missing opportunity to benefit society. See, both the things are there. By the new drug, it should not be harmful. And if it is really a potent drug, you should not miss that. Okay? If drug known to be better, there is no control group. If drug known to be better, no control group. Alternative treatment as a control. Say, when do you go for a placebo? Say the dictamis is when there is nothing available, then you go for a placebo. If there is even some potent drug is available, you will have to rather give. Say, give the available best to your control group. Okay? Available, known best to your control group. Efficacy of the drug must be truly unknown. Suppose you already know it is 90% efficacious, you don't go to do the controlled clinical trial for that. Okay? Yes, sir.
I would like to refer to the paper by one guy called Miller, Placebos, the Ethics of Placebo in Psychiatric Research, published 2006. I don't remember the journal or journal name, where he says 2 claims, because I have no idea whether or not they are true. You will be in a better position to comment on it. FDA claims that placebo trials are needed to show efficacy incontrovertibly, unambiguously. Particularly in psychiatric research, I believe that even if your test drug is shown to be better than the standard of care, it could still be doing only as well as placebo because of positive placebo reactions. And therefore, FDA insists, particularly during the maintenance phase, not during the acute phase though, placebo-controlled trials, in spite of the fact that, so the trials have started having a 3rd arm with placebo. So you have a standard of care, placebo, and...
Excellent. What you said is not a question, it's a, see, there is an effect called a placebo effect. Okay. Sometimes, you can even see it in the clinical practice. The doctor may give just a sugar pill, and then say it's an excellent tablet that is available, just now they have proved that it is very good, I am giving it to you. And if after taking it you may feel better. See, that's a feeling that somebody is carrying, and this is more so in a psychiatric situation. Okay, so you are right. So placebo could be used in, that is a big controversial stuff, and there is no standard global answer for that. It should be more on a specific issue or where you are dealing, whether it is applicable or not. Okay.
Sir.
Yes, sir.
Your previous slide.
Yes.
It is number 2. If drug known to be better, no control group. Please clarify it.
Yeah.
In what sense is it to be known?
No. If there is a better known drug available, in fact, the wording is not all right now. If there is a better known drug available, there is no control group required. So you give that known better drug. Okay. I understand that wording is not very clear there. Informed consent, this is again, there is a huge issue on this also. See, when do you inform the people? Before randomization or after randomization? And how are you going to say, in a normal circumstances, what we do it is say in the government chest clinic, if somebody is having all eligibility criteria, then he is sent to ICMR. Okay. In ICMR, they try to explain him the whole trial. They say, this is the existing thing, that is a new thing that come, found to be safe. Okay. But we don't know whether it is better than this or not. We feel that it is better, but there is no proof. So we are going to rather do, some people we are going to give the existing things. For some people we are going to give this new drug, and at the end we are going to see whether this new drug is better than that. Okay. And if you are willing to join us in this trial, we will be very happy. You may either get the standard one or the new one, which we don't know. You also don't know, we also don't know. Okay, you may get one of these two. At the end, we will see whether this is better or that. Are you willing to come and join for this trial? Roughly speaking, this will be the sort of an informed consent like. Okay, in the local language, in a way, a lay person can understand, it's not very scientific and things and all. Okay? And suppose the person says yes. Then you get his signature, some witness signature and all those things. Then you also tell him, you are joining here, doesn't mean that you have to rather go through the whole procedure. In between, if you want to withdraw, you can withdraw. And we won't ask you any reason why you are withdrawing. If you want a standard treatment at that time, we will not deny you the standard treatment. We will give you all these promises we are supposed to give. Okay, in an informed consent. Okay. After he says yes, then we can have the random situation, random allocation of them. He may either go to treatment A or treatment B. One of them are test, one of them is standard. So, but there are certain methods where, even after randomization, there is, he has given an opportunity. It's called, the whole trial is called Zellen's trial. He has come out with different variations on this, wherein switch over from this to that is possible, but still it is taken as intent to treat analysis to be done. These are all some of the complications of the RCTs, an extension of the routine RCTs. Let's not bother about it too much here. Okay, the stopping rules to be stated before, and the stopping rules actually couldn't be based on the clinical efficacy of the test treatment. Both ways, if the test treatment is horribly bad, you stop. Or if it is extremely good, then also you stop, because it is unethical to give a lower potent drug to 50% of other guys. Okay, so if it is really good in the interim analysis, you stop the trial, and this becomes a standard therapy later on. Okay, toxicity of the test treatment, you have to clearly state in the protocol, when do you call a drug as toxic? Okay, what level is admissible and things and all. And usually in these sort of trials, there will be an expert committee, which would be an external expert committee to look at, because say if I do a trial and I am an expert, and if I alone look at it, I may close my eyes on certain things, so it is always better some external guy comes and does it. Bias, as I was mentioning, incomplete review of literature is also contributes to a selection bias. But say the last publication bias, this is also quite an important stuff when you are going to do a systematic analysis, review. What happens, usually people, they publish only if there is a significant difference. I have seen number of guys, I did this work, sir, and unfortunately, statistically it is not significant. They tie it around and put it in the loft, and they don't publish it at all. Okay. So what happened? Most of the published records would be a record which are positive ones. So the negative things you may not rather get at all. Very few negative things are published. Even if you are the negative and then you send it for journals, some of the journals may not be too keen or too interested to publish some negative results. Okay? So there is a publication bias, especially when you are going to do some systematic review. This publication reviews comes in. So in conclusion, RCT is the standard, but not the only approach. Rigorous scientific assessment, it's absolutely essential. So involve researchers and practitioners in the concerned system of medicine. Suppose we are doing a traditional medicine research. For every stage we involve the people who have the traditional medicine stream also as part of the trialage. Ethics and human rights play a huge vital role in RCTs. Okay?
Is there is it a standard format for RCTs? Is there an established format for reporting SAEs?
Yes, there are. And see, again, this sort of an adverse effects also varies between the type of investigations that you are going to rather do. And it also depends on what's your primary interest, and suppose in many trials your primary interest itself is to look at the adverse effects. Okay, so there is guidelines available as how to rather go about it, but that's again be very specific to your trials that you are going to rather do on it. Okay, yeah. And they say there are certain limits within, if it threshold level, if it crosses that, then, even you can stop the trial at that stage.
So one more question regarding the stopping rules. You said that it should be like some independent set of observers should be there for that kind of an analysis.
Not only for that, the whole conduct of the trial, there could be observers.
So who should comprise of this independent
that is put
set
in
of
protocol
observers?
Earlier. If these are all the events A, B, C, D happens, I am stopping. That is, you are deciding even before you conduct the trial.
So my question is, who would be the observers? I mean, would they be statisticians, epidemiologists, researchers, or ethicists?
Group of them, yeah, maybe a group of them. See, monitoring committee. In fact, large trials they have different technical monitoring committees. Like some committee they look on the, say for example, there was some sort of a huge vaccine trials, Malawi trials. Our director, Dr. Gupta, was a part of the monitoring committee to look at the potent of the vaccine. So they know, you really do not know whether different batches of the vaccine, they are equally potent and things and all. So they take a sample of those batches, they send for assays and see the content of it. So the monitoring committee can also have different roles and responsibilities. Different people may look at different angles of it. It all rather depends on the intensity, the size of your trial. Some small clinical trial you do on 100 patients may not be a huge stuff, but there are certain huge, large trials which are done, got an international monitoring committees.
So all these should be clearly mentioned in the protocol.
Yes. And protocol is a sacred document, Bible. Okay, you can't rather change them. Protocol is the one, and one committee, central statisticians mostly are involved in adherence of protocol, is usually entrusted to statisticians because clinicians may have some inherent bias. Some patient comes, he may try to rather say this is a better drug, why can't I give him? Because his medical ethics, would rather, if he has a feeling that this is a better drug, he may try to rather give, he may manipulate. So a statistician is as a central guy who doesn't rather fully involved, as not a principal investigator, is usually entrusted the job of monitoring the protocol adherence. Okay, thank you very much.